Hello friends, in this video we will talk about the transgenic animals, their definition, their procedure to form the transgenic animals, and we will talk about the importance of transgenic animals. In the next video we will then talk about some examples of the transgenic animals. Now first of all, what is a transgenic animal? A transgenic animal is the one whose genome has been altered by the transfer of a gene or a genus from another species or breed. So a transgenic animal is the one which carries a foreign gene, and that foreign gene or foreign genes, they can come from the another species altogether, or they can come from the same species but of a different breed. So that will be a transgenic animal. Now the first transgenic animal that was developed were the mice. So mice were the first transgenic animals. First transgenic animals. Now this transgenic mice was first developed in the year 1985. So from nowadays a number of transgenic animals have been developed. For example, nowadays there are transgenic sheep, there is transgenic even goat, there are transgenic rabbits, there is transgenic cow, there is transgenic dog, and even there is transgenic monkey. But of all the transgenic monkey in these animals, the 95% are transgenic mice alone. So 95% of all transgenic animals that have been developed so far, 95% of transgenic animals are mice alone. They are mice alone. So most numerous of the transgenic animals, they are the mice, 95%. Now transgenic animals, production of transgenic animal is a multi-step process that can be briefly summarized here. So we will talk about the steps involved in the production of the transgenic animals. So steps involved. Transgenic animals, for the formation of the transgenic animals, first step is to isolate or to select the desirable gene. So first step will be the selection of the desirable gene. First, the gene to be introduced into the transgenic animal is to be decided. So once we have decided what type of gene is to be introduced, then we can go for the next step, that is the isolation of the desired gene. So next step will be the isolation of desired gene. So once we have isolated the desired gene, then we can amplify this desired gene, and amplification can be carried out using bacteria or more commonly by using the polymerase chain reaction. So the third step will be the amplification of desired gene, and this can be carried out by bacteria. This can be carried out by bacteria, or it can be also carried out by the most popular reaction, that is the PCR or polymerase chain reaction. That is polymerase chain reaction. Now the next step is now the introduction of this foreign gene into the host. Now for this purpose, there are two options are available. In the first option, as I can show you in the image here, in the first option, what we do is that first the animal, the females of the animal whose genome we want to modify, that is made to superovulate. So first the females of such animals, for example here is the transgenic mice, so where first step will be that the females will be superovulated. Now this step is carried out by administering some hormone based or the FSH based drugs to the female. So this will result in superovulation, which means that multiple gametes will get matured, and this mice will produce many such gametes. Now after that, such gametes are harvested. The gametes or the female eggs are harvested from the mice, and then in vitro fertilization is carried out outside the body of female. Now after the fertilization, before the syngamy, before the male pronucleus, as you can see the bigger one here, the bigger nucleus here is the bigger nucleus is the male pronucleus, and the smaller one is the female pronucleus. Now before a male pronucleus fuses with the female pronucleus, a process which is called as syngamy, then the male pronucleus is inserted with the transgene, and this transgene is inserted with the help of a micro injection. So here is a micro injection or the micropipette. With the help of this micropipette, the transgene or the desirable gene is inserted into the male pronucleus. So here this is transgene is being inserted into the male pronucleus. Now after that, the male pronucleus will fuse with the female pronucleus, and that will result in the formation of zygote. And then that zygote is then allowed to undergo multiplication, and after few divisions, for example after 8 cell stage or blastocyst stage, then that embryo is implanted in a surrogate mother. So here you can see, then these embryos, they are implanted in a surrogate mother. So this is not the actual one from which eggs were developed. Then these embryos, they are implanted in a surrogate mother. So they are to be inserted or implanted in the surrogate mother, and then the surrogate mother will give rise to the new offspring that is born will have the transgene in its all cells. So all these transgenic all the offsprings will therefore carry this gene, and they will be called as the transgenic animals. So transgenic animal will be formed. Another procedure, alternate procedure, instead of using the micropipette, we can make use of certain viruses, and that procedure is explained below. So in this image you can see that here the donor female, from the donor female 8-celled embryo is harvested. So first 8-celled embryo is harvested, and this 8-celled embryo, the transgene is inserted with the help of a retrovirus. So with the help of a retrovirus, a transgene is inserted at the 8-cell stage. In all the 8 cells, a transgene will be inserted, and then the 8-celled embryo will be implanted again in the surrogate mother. Then the offspring that will be produced will again have the transgene, and it will be called as transgenic animal. So this is about how transgenic animals are produced. So the next step in the production of transgenic animal will be the transfer of the transgene to the host. So that will be the transfer of the desirable gene into the host using either viruses, especially the retroviruses, or micro injections, right? So this was the summary of how these transgenic animals are formed. Now next we will talk about what is the importance of these transgenic animals. What is the purpose of forming these transgenic animals? Now transgenic animals are developed or formed for different purposes. Some of them are as follows. So we will talk about now the importance of transgenic animals. Now, the first importance of the transgenic animals, the transgenic animals are developed for several reasons. Now, one of the reasons is that they are used to produce the human pharmaceuticals. So, some of the complex human proteins are developed by these transgenic animals by introducing the genes of those proteins into the transgenic animals. For example, there is one protein that is called as the alpha-1 antitrypsin. Alpha-1 antitrypsin is a protein that is used for the treatment of a condition which is called as emphysema. Emphysema, if you know, that it is a disease of the lungs. So this emphysema, it can be treated by alpha-1 antitrypsin. It is a protein. Therefore, this gene for antitrypsin, alpha-1, gene is introduced into the animal. It is introduced into the animal, which will then produce this protein. Animal will produce the protein, and then that protein is harvested. It is harvested, and then it is given to the patients for treating this emphysema. Similarly, another example of the pharmaceuticals that can be developed, pharmaceutical compounds that can be developed by these transgenic animals includes the CFTR protein. And we know that this CFTR protein, this is responsible for, it is faulty nature, is responsible for causing a disease that is cystic fibrosis. So again, this CFTR can be corrected. This cystic fibrosis transmembrane regulator, regulated protein can be harvested from the transgenic animals. So the first application of the transgenic, or importance of the transgenic animals, is that we can obtain the pharmaceutical chemicals from them. So they are used for the synthesis of the pharmaceutical compounds. Pharmaceutical compounds are the compounds which are used for treatment of the disease. Number second importance of these transgenic animals is that transgenic animals can be made hypersensitive. So once we make them hypersensitive, we can then study the vaccine safety on them. So before trying the vaccines on the humans, they are first tested on the animals, and transgenic animals can be made hypersensitivity to study whether the vaccine that has been developed is safe or not. So number second importance of transgenic animals is that they are developed for the vaccine safety. So vaccine safety, for the purpose of studying the vaccine safety, transgenic animals are produced. Number third importance of the transgenic animals is that they are also used for the studying the chemical safety. Now there are certain chemicals that are known to damage, or that are known to cause harmful effects on the genes. Therefore, what is done is that genes are exposed to these chemicals. Genes are exposed to the chemicals. These are exposed to the chemicals, and then such exposed genes, then the exposed genes are transferred into the transgenic animals. Then these are then transferred to the animals, which will now be a transgenic animal. So now it will become a transgenic animal, and then this transgenic animal is then studied to find out what kind of effects has the chemical produced on the gene, and what kind of harmful effects are those of the chemicals. So before chemicals are they are first tested on these transgenic animals. So number next importance of the transgenic animals is that these transgenic animals are used as disease models. So this is the number next importance, that transgenic animals are used as disease models. So in order to study the disease, how disease develop, how they can be treated, transgenic animals are developed. So most of the mice especially, they are used as a disease model, and there are certain human only disease. So certain disease, especially the genetic disease, they occur only in the human. So on human only disease can be developed in the mice or in the transgenic animals, and then their course of development, their progression and the effects of the medicines on them or the drugs on them, that can be checked. So they can be used as disease models for studying the disease, for studying the human only disease, for studying the progression of disease, as well as how these disease can be cured or how these disease can be prevented. So this was about another importance of the transgenic animals. Another importance of transgenic animals is that we can study them for the gene expression. So these can be used for studying the gene expression. For example, we don't know what is the exact function of the gene in human beings, so we can, of a particular gene in human beings, so we can isolate that gene and introduce the gene in the animal, a transgenic animal, and then study its effect, and then study and then observe the transgenic animal to find out what kind of expression that gene shows in the transgenic animal. So this is used how particular gene is controlled, how particular gene is expressed. So that is another application of the transgenic animals. Another importance of transgenic animals is that they can be used as the superior parts. So they can be used to give us the superior parts. For example, we know that pig heart is closely related with that of human heart. So it has been observed that pig heart is closely related with the human heart. So with the human heart. So these organs, human heart. So there are thousands and millions of the patients which are awaiting these organs, who need this organ transplantation, but organs are not available. So for them, these animals can be made, the MHC molecules of the animals can be masked. For example, the MHC cytus of the pig can be masked, and then its heart can be harvested and can be inserted into the humans who need them. So they can be used. Similarly, other organs like kidneys and other organs, they can also be harvested from the these transgenic animals by first masking their MHC cytus. So here MHC cytus of the MHC cytus of the transgenic animal can be masked, and then organs can be harvested, for inserting into the humans who need them. Another importance of the transgenic animals is that they are developed for the food purpose. So transgenic animals, they are used for developing better quality, better quantity, or better size of the food. So they are transgenic animals, they have been found, they are developed for the food value enhancement. So their food value enhancement. Food value enhancement means that the food, for example, they will be of greater size. For example, fish, a transgenic fish will be of greater size, or it is meat will be having more nutrition, or if it is a milk animal, then the quantity and the quality of the milk will be higher. So that is, they will be having the size will be bigger or than the normal, or their quality is improved. So these are the some of the reasons or some of the importances of the transgenic animals. So in the next video, we will now talk about the some examples of the transgenic animals. Hope you liked the video. If you like it, please give it a thumbs up and do share and subscribe the channel. Thank you.